levels of control. So metabolic processes, remember metabolism is a whole series of chemical reactions. So metabolic pathways are a series of chemical pathways, and they're always dependent upon biochemical catalysts. And what are the biochemical catalysts? Enzymes and ribozymes. Don't forget the ribozymes. Enzymes and ribozymes. So a cell's ability to make these catalysts is dependent upon what? The whole topic we were talking about last time, or that I was talking to you about last time, which is DNA. DNA is the genetic information of a cell. The nucleotide sequence of DNA determines the nucleotide sequence of RNA, whether the RNA is a ribosome or the RNA is a messenger that's telling the cell what protein to make. It's always DNA that determines what type of catalyst the cell can make. So DNA, and specifically regions of DNA called genes. Although we'll come back to that because the definition of a gene has changed a lot over time. So whether or not a cell gets to use its metabolic processes is determined by a bunch of regulatory mechanisms. And there's regulation at every level. If you think about what's going on in the cell, there's regulation at the level of metabolism, so there's regulation of enzymes. There's regulation at the level of transcription, where RNA is being made. There's also regulation at the level of translation. The good news for you guys is we're only looking at two. So we're looking at two mechanisms for control. Well, except I'm going to sneak in the CRISPR again, but it's still, anyway. So we're looking at control at the enzyme level, and then we're looking at control at the gene level. The enzyme level is pretty easy. So enzyme level control is referred to as feedback inhibition or end product inhibition. Key word here is inhibition. So when I was talking about how you regulate enzyme activity, we talked about a variety of factors that can influence enzymes, and I talked about inhibitors, allosteric inhibitors, and competitive inhibitors. It's critical that you recognize that if you see the word inhibition, you're talking enzyme level. And so when we talk about gene level regulation, there'll be a different word that applies. It's not inhibition. So feedback inhibition, end product inhibition, it's enzyme level control. This involves allosteric regulation. What's the difference between allosteric and competitive?
One binds at the activity site and the other one doesn't.
Which one?
The allosteric does not.
So allosteric, remember allo means other. So allosteric inhibition is inhibition that is applied where the molecule that is the inhibitor is binding to a site other than the active site. It's not binding to the active site of the enzyme, it's binding someplace else, and in the process it changes the active site of the enzyme so it doesn't work anymore. So yeah, anyway, there's a picture somewhere, here it is. So in this picture, the allosteric inhibitor is this little guy down here. It's binding to this site, this is the active site on the enzyme, and when this little allosteric inhibitor jumps in there, it changes the shape of the active site, and now this substrate can't bind in there anymore. So allosteric, again, it involves an inhibitor that's binding someplace other than the active site. And that's how feedback inhibition is going to work. Now, the reason this is called end product inhibition is because it's typically the end product of some metabolic pathway that inhibits the enzyme. So it's coming from the end of some metabolic pathway, and it's going to go, and it usually acts on the first or second enzyme in the pathway. So I'm going to give you a specific example. Oftentimes what you get presented is something that's in a lot of biology textbooks. And so this one is biosynthesis of isoleucine. Isoleucine is an amino acid, and amino acids are made oftentimes from other types of substances. So in this case, this pathway to build isoleucine involves threonine, which is also an amino acid. And there's 5 steps in the metabolic pathway, which we're going to say that the 5 steps are catalyzed by enzymes. You don't have to know the names of these enzymes. We're just going to call them enzyme A, B, C, D, and E. So there's 5 steps. So the metabolic pathway looks like this, where threonine, an amino acid, gets converted into substance W, and W gets converted into substance X, and X gets converted into substance Y, and Y gets converted into substance Z, and Z gets converted into isoleucine. So it's a multi-step process. Each one of these arrows represents a chemical reaction that's occurring, And the chemical reaction is catalyzed by a specific enzyme. So A catalyzes this reaction, B this one, C this one, etc. Isoleucine is the end product of the pathway, and it's also the inhibitor. So what it does is come back and act on this enzyme, enzyme A, and blocks the activity of that enzyme. Again, it's going to change the active site, so the active site of the enzyme can't bind threonine, consequently the pathway is shut down. So again, the letters W through Z represent metabolic intermediates. Those are things that are made in between. We're only mostly interested in isoleucine, which is our allosteric inhibitor. And again, typically the end product will inhibit either the first enzyme or the second enzyme of a pathway. It's going to shut this pathway down early on. But can you see this as a highly efficient way to control metabolism? Think about what goes into making enzymes. The cell has to open up the DNA, it has to transcribe the DNA and make messenger RNA. Every nucleotide that's added when you're building messenger RNA is equivalent to 1 ATP. So there's a lot of energy being used there. Then, after you've got the messenger RNA, you have to do translation, which means you're binding a bunch of amino acids together. Again, every binding, every peptide bond, that's another ATP. So the cell is expending a lot of energy making messenger RNA and then making protein, and the pathway is just shut down by allosterically inhibiting the first enzyme. It's a quick regulation, and it's also reversible, because if the concentration of isoleucine is decreased, it'll fall off the enzyme and then the enzyme can be activated. But it's not very efficient. This is sometimes referred to as negative feedback. The negative feedback, anytime something happens that then has a turning off effect, which this does, it's going to shut the pathway down. That's considered negative feedback. It would be positive feedback if it made the pathway work better, which it doesn't. But again, it's not terribly efficient. Both eukaryotic cells and prokaryotic cells can use this. It's a rapid acting mechanism, and as I said, it's reversible. But it's not energy efficient, because the cell has to expend so much energy making messenger RNA and then making a bunch of proteins, those enzymes, that it's not going to use. Consequently, there's another way to control. So that's just saying the same thing I just said. A lot of energy expended. This is a review. So what term applies if you're going from DNA to messenger RNA? Transcription. Rewriting genetic information, nucleotide sequence to nucleotide sequence. And if you're going from messenger RNA to polypeptide? Translation. You're changing languages, going from nucleotide sequence to amino acid sequence. Don't forget the meaning of those two words. Also, don't forget replication. So transcription occurs where? Wherever the DNA is, right? In the nucleus, in the nucleoid, in association with plasmids, in association with the DNA in mitochondria and chloroplast, wherever there's DNA. And it requires DNA as a template, enzymes, and energy, DNTPs and RNTPs. No, only RNTPs if we're doing transcription. Translation occurs where? Ribosomes. And what's required for translation? You've got to have messenger RNA, you've got to have ribosomal RNA, you've got to have transfer RNA, and the enzymes associated with those. And you have to have energy. So the ribosomes, the ribozyme, which is peptidotransferase, got to have codon to anticodon hydrogen bonding, all kinds of fun stuff. Yeah. So, getting a metabolic pathway up and running then requires a great deal of work. Again, the cell has to expend a lot of energy just getting enzymes ready to use. And then a lot of other review here. What kind of enzymes are essential to cell function, so they're always being made? Constitutive. This is taxing your brain, it's not, because you just have to download a bunch of information on the lab exam. So, Constitutive enzymes then are always being made, always being used. And when I was talking about categories of enzymes, and I said, we're gonna look at constitutive, that was quite some time ago. And I said there were two other categories that we weren't gonna talk about yet because it wasn't gonna make any sense. Anybody remember what they were? Repressible, and inducible. So now it's later and we can talk about repressible enzymes and inducible enzymes. So repressible enzymes are enzymes that are encoded by genes where the transcription can be turned off. When you block transcription, you don't call it inhibition, you call it repression. So if it's gene level control and you're turning something off, it's repression. If the enzymes are inducible, they're regulated by, or they're encoded by genes that can be turned on. What's being turned on is transcription, and so those genes are called inducible genes, and the enzymes associated with those are inducible enzymes. So we're going to talk about repressible and inducible systems. What do you think is meant by gene expression? A gene is a piece of DNA, so if a gene is being expressed, what does it mean? It's being copied into messenger RNA, and the messenger RNA is going to the ribosome, and it's being translated, usually. However, that meaning has changed somewhat because it used to be assumed that gene expression meant you had to get a polypeptide, and since we now know that some RNA are catalysts, as long as you get transcription accomplished, you've still expressed a gene. But typically it's DNA to messenger RNA to polypeptide. So now we're going to look at genetic control, regulation exerted at the gene level. And again, I'm going to give you a couple of examples. So in prokaryotic cells, genes that encode metabolic pathways are typically located within a region of DNA. You'll have a whole series of genes encoding a whole series of enzymes, and those enzymes are going to run a metabolic pathway. And for a while, this was kind of speculated, but it wasn't absolutely proven. Now it's definitely been proven. So there are these regions within prokaryotic cells that are called operons. An operon is DNA. Specifically, an operon is a series of structural genes that are going to regulate, or that are going to encode enzymes to run a pathway. So you have a series of structural genes. You have regulatory regions of DNA. One of those is the promoter. Another is called the operator. We haven't talked about operators yet. And there's one called an attenuator, which I'm not going to talk about. And then I gave you a term when we have multiple genes that are copied into one long messenger RNA, which is, again, pretty characteristic of prokaryotic cells. They will usually not express genes individually. They'll express genes as a group. And so the messenger RNA that they make is the product of what we call something transcription. Anybody remember? The term is polycistronic. Polycistronic. So it's polycistronic transcription. Oh my, polycistronic. So an operon, again, it's a segment of DNA. It's a series of structural genes and the control elements that regulate the transcription of those genes. Most important to remember as we go through this section, what's DNA, what's protein? So an operon is DNA. It's genetic material. Again, pictures of DNA, so you get that picture. The control elements associated with operons, the promoter site, the operator site, sometimes an attenuator site, but you guys don't have to know it. These are also DNA. So the promoter site, do you remember what the promoter site is for? The function of the promoter? Remember when I was talking about composition of RNA polymerase and there was that big pink section? And I said that big pink section is really part of RNA polymerase, but it doesn't stay with the core enzyme. It goes to a specific site on the DNA and says, this is where the work is going to get done. And the work is transcription. So the promoter site is where sigma factor binds. Sigma factor is that special part of RNA polymerase, the foreman, if you will. Sigma factor binds to the promoter. The promoter then is where transcription can start. It's like the start point. Sigma factor of RNA polymerase has to bind there. So the promoter sigma factor interaction, that's basically going to determine where transcription is going to go, where on the DNA it's going to be. So it could be on one strand or the other. It's sigma factor binding a promoter that determines where that process is going to take place and in which direction it's going to go along the DNA. This next one we haven't talked about yet. So the second site is called an operator site. And you can think of an operator site as being an on-off switch. It's on or it's off. When we say something is off, it's repressed. When it's on, it's not repressed. But the key is, what are we controlling? Since the operator is part of DNA, DNA has to be what in order to get a product? Whether you're talking about making ribozymes or making enzymes, there's a process that has to happen with DNA, and it's not replication. The process is transcription. Okay, so operator site is on-off switch, and what's being turned on and off is transcription, making RNA. So the operator site gets turned on and off by these proteins called repressor proteins. The repressor, if the repressor binds to the operator site, then transcription is off. If the repressor isn't bound, then transcription is on. That's why we say it's like an on-off switch. But it functions with these repressors binding or not binding. The thing about repressors is sometimes they're active alone, and sometimes they're not. So they're kind of like enzymes when we were talking about conjugated enzymes and simple enzymes. So if a repressor is active alone, it's kind of like a simple enzyme, or it can function all by itself. And if it's not active alone, then something else has to bind with it, and that something else is referred to as a co-repressor. So we're going to look at two examples where repression can be exerted or where induction can occur. Sometimes an operon is off unless it's induced, which just means the repressor is normally on that operator site, and you can't get transcription to happen unless you get the repressor off. So that has to do with whether the enzyme, whether the repressor is active or not. Does it have to be induced? Does it have to have a co-repressor? So again, we're going to look at some specific examples. The other thing to remember is that most operons are constitutive. Most enzymes are constitutive. If they're constitutive, there's no operator. You can't turn them on or off. There's a promoter, and that's it. And as I said, the attenuator, that's another complication of the system, and we're not going to worry about it. So we're going to look at two examples. Hopefully you guys can hang in here with me through two examples. So the first one has to do with tryptophan biosynthesis. Tryptophan is another amino acid. So we're going to be looking at a metabolic crossway, a metabolic process or metabolic pathway that's involved in tryptophan biosynthesis. This is controlled by a repressible operon. So a repressible operon is one that can be repressed. That means it can be turned off. So we're going to be looking specifically at tryptophan biosynthesis in Escherichia coli. This is usually what happens in biology. There will be some pathway that is identified, and then it gets incorporated into textbooks, and then everybody uses that as an example. So this is a specific example of a repressible operon. The way this works, you have two substrates, charismatic acid and glutamine, and the cell runs a metabolic pathway. Again, this pathway is going to involve 5 enzymes. The enzymes are A, B, C, D, and E. You take these two substrates, charismatic acid and glutamine, you go through a series of intermediates, and you get to tryptophan. So tryptophan is the end product. Hopefully by now, when you see a metabolic pathway sketched this way, it kind of makes sense. But it's not like the glycolysis pathway where we named a bunch of intermediates. In this case, we're just putting letters on. So we know there's a series of steps, there's a series of intermediates, And we don't really care that much about that. So again, charismatic acid and glutamine are the substrates. The ones, the letters W through Z are these intermediates, and tryptophan is the end product. If we were still talking about allosteric inhibition, tryptophan could potentially inhibit the first enzyme in the pathway. But we're not talking about enzyme level inhibition. We're going to be talking about genetic regulation here. So this pathway is going to be controlled by an operon. The letters A through E are enzymes, and those enzymes have to be catalyzed by, or excuse me, have to be encoded by genes. So the genes that encode the enzymes A through E are all on this tryptophan biosynthesis operon. So again, an operon is a segment of DNA. This is a diagrammatic representation of the DNA, so I've made it purple to remind you, yeah, we're talking nucleic acid here. These genes, gene E, B, C, B, and A, is what they're technically called. Notice that they're not in any, well, they're backward. So A, B, C, D, and E, read backward. This is the promoter site, where sigma factor binds. This is the operator site, which is the on-off switch. What turns that off is a repressor, which is a protein. So a repressor protein has to be encoded by some other gene. The gene that encodes the repressor isn't part of this operon. So this operon just has the promoter site, the operator site, and a series of structural genes. And again, keep in mind, the operon is DNA. The other thing you have to keep in mind is I show you a metabolic pathway. I say you've got enzymes, A, B, C, D, and E. And that the enzymes are encoded by these genes, A, B, C, D, and E. What has to happen between genes and enzymes? Transcription. You have to make RNA. So don't forget about that, because what we're really controlling is transcription. So here's our operon. The promoter site again is where sigma factor binds to begin transcription. So sigma factor attaches there. The operator site is where the repressor can bind, and the repressor is a protein. If the repressor protein is bound to the operator site, you can think of it as being a roadblock. And again, the gene that encodes the repressor is not part of that operon. It's located in a different region. So that's the E. coli chromosome. I just put an arrow on there randomly and said, here, it's somewhere there. I have no idea where it is. It's not part of the operon. It really doesn't matter to us where it is. There's another gene that encodes the repressor. The repressor is a protein. We don't care. Key to this repressor is that it's a protein that's kind of like a conjugated enzyme. By itself, it's inactive. So it is an inactive repressor if it's by itself. It requires a co-repressor. So under most circumstances, sigma factor can bind to the promoter of the tryptophan biosynthesis operon. And when sigma factor binds, then the core enzyme binds, and then you see that movie with the RNA polymerase zipping down the DNA and the RNA being made fast as can be. So transcription is going to proceed. Most of the time, transcription can happen. Again, sigma binds the promoter, there's nothing to stop it. And so sigma binds, then the core enzyme binds, and then whoop, transcription. We're going to transcribe that whole series. We're going to get one messenger RNA that is a copy of all 5 of those regions of DNA we're calling genes. So they're all transcribed together. Transcription is polycistronic. Repetition is learning. I know it's tough because your brains are fried. Okay, so again, while the operon is active, transcription is happening, we're going to transcribe all those genes. We're going to make one long messenger RNA that has all the information to encode those enzymes. And we need the enzymes to run the pathway. The translation, on the other hand, doesn't happen all at once. Instead, you get ribosomes jumping on at different specific start points, so that when translation happens, those pieces of RNA are going to be used to encode polypeptides more or less separately. You're going to have a bunch of different ribosomes involved. But if the cell is allowed to produce tryptophan for a long time, and it gets to the point where there's enough tryptophan being made, let's say this cell is now kind of slowing down, it doesn't need to keep making tryptophan, it wants to stop tryptophan biosynthesis. When we have a lot of tryptophan, instead of acting as a repressor, instead of acting as an inhibitor, it's going to act as a co-repressor. So what it's going to do is bind with the inactive repressor protein and make it active. It's going to activate it. And then that tryptophan repressor complex is going to bind to the operator site. So here's the repressor by itself. By itself, the repressor can't do anything. We attach tryptophan to it, and the whole thing is going to jump onto that operator site. My animation is a little lame, but anyway. So there's the tryptophan repressor complex. It binds to the operator, and now, as you can imagine, even if sigma factor attaches to the promoter, it can't go beyond that point. It can't go ahead and transcribe the DNA. So nothing's going to happen. Transcription is blocked. And as long as that repressor tryptophan complex is bound on there, transcription can't occur. The good news is this is concentration dependent. As the amount of tryptophan in the cell begins to dwindle, as it decreases, then tryptophan will let go of the repressor, and then the repressor falls off the operator, and transcription can go again. So it's not permanently off. This is an extremely efficient mechanism, because if the cell doesn't need to produce the end product, tryptophan, it doesn't just shut down the metabolic pathway, it shuts down the transcription at the DNA level. We're not going to transcribe, we're not going to expend a bunch of energy making messenger RNA, we're not going to expend a bunch of energy making proteins that the cell doesn't need. So it turns it off at the gene level. The other thing is there's a little disclaimer up here. This is an incomplete description of tryptophan biosynthesis, because there is an attenuator on this operon, and I'm not talking about it. If you want to go look it up, feel free to. It just complicates the situation, and it's complicated enough. So I want you to understand the tryptophan biosynthesis is repressible, which means transcription is normally go, it's normally on, but it can be turned off, and if it's turned off by a repressor binding to the operator site. The repressor can only bind if tryptophan is in high concentration, because the repressor requires tryptophan as a co-repressor. So what's really significant about a repressible operon is typically you have a repressor protein that's inactive by itself. You have to bind something with it in order to activate it, and then it will stop transcription. The other thing is that not all bacteria control the same metabolic pathway the same way. So this pathway is specific for Escherichia coli. If you were to ask, do all bacteria regulate tryptophan biosynthesis this way? The answer would be no. Well, bacteria don't all even produce tryptophan. Anyway, the next example that I'm going to give you is an example of operon controlling lactose utilization. This is sometimes referred to as the lac operon, and when it was first diagrammed and included in references, there were only 2 genes associated with it. Now there's 3 genes associated with it. This is an inducible operon, and an inducible operon is one that's off, but can be turned on. What's off and can be turned on? I say the operon, but it's actually what process occurs at the operon. And the process that occurs at the operon is transcription, because we're talking about gene level control. So lactose utilization in Escherichia coli. Again, E. coli is often used as the model because it was an organism a lot of people were studying, and so they could really look at the genetics and know what was going on. Lactose utilization is controlled by an operon that is usually off. It's usually repressed, and this makes sense if you think about it for a little bit, because what is lactose? Sugar, but more specifically, lactose is milk sugar. Milk sugar is only available to E. coli under certain circumstances. Take cattle, for example. But you could talk about cows, horses, sheep, rabbits, humans. When is lactose available to E. coli? E. coli lives in the gut. Think about the lifespan of, your friendly neighborhood horse. I don't know how long a horse is. Huh? Yeah, infancy. So you've got E. coli living in the gut of an animal that only has lactose available to it when it's young. Because lactose is milk sugar, and those animals are only drinking milk for a short period of time. But the E. coli are living in the gut of that animal for years and years and years. Again, the E. coli want to be efficient, so most of the time their lactose utilization operon is off. There's no lactose to be had. Adult cattle, adult horses, adult rabbits, dogs, cats, whatever, they don't drink milk. They eat grass, they eat leaves and hay and so forth. So most of the time this operon is off. Again, it's a matter of efficiency. But it can be turned on. And again, what's being turned on? I can say the operon is turned on, but what's the process that gets turned on? Transcription. Ah, so you're getting this. Feel like you're being browbeaten. Transcription. Okay, so this lac operon, there's 3 structural genes, there's a promoter and an operator. It turns out there's actually 2 promoters. So this operon has a gene up front called the LAC-I gene, and that LAC-I gene encodes a repressor. Notice there's a promoter, and then the LAC-I gene, and then there's another promoter followed by an operator. So if you have an operator, that means transcription can be turned on or off. But if you don't have an operator, there's a term that would apply to this, what this gene encodes, because it cannot be turned on or off. Consequently, the protein that is encoded by that particular gene is constitutive. You can't turn that transcription on or off. So we have the LAC-I gene. The LAC-I gene is going to encode a repressor. That LAC-I gene encodes a repressor that is constitutive, which means it's always being made. Furthermore, it's active alone. So as long as that operon is intact, if sigma factor binds to the promoter that allows the LAC-I gene to be transcribed, then the LAC-I protein is going to be produced, and that is a repressor, and it's going to bind to that operator and turn that LAC operon off, because it's active alone. So the repressor is constitutive. That's why under most circumstances, the structural genes associated with this operon are not going to be transcribed. There's our repressor sitting on the operator. Again, it's like a roadblock. As long as that thing is on there, there's no transcription of the LAC A, Z, and Y. Not sure exactly why those letters are applied, but anyway. And again, it's just because E. coli that's living in the gut of an adult animal doesn't have lactose available. That's not true of humans because we eat all kinds of weird things. But if you're talking about an adult cow or an adult deer or an adult horse, they don't drink milk. And they can live for years. So their E. coli have to be efficient. So then we can talk about those structural genes, the LAC, Z, Y, and A. Those structural genes encode enzymes that have to do with lactose utilization. Again, I'm not sure why the letters are applied the way they are. LAC-Y encodes an enzyme called beta-galactoside permease. In old references, it was always referred to as beta-galactoside permease. More recently, they just said, let's just call it lactose permease. Do you know what a permease does? Because in lab, we were talking about citrate permease. Remember what citrate permease does? It brings citrate through the cell membrane. So lactose permease also brings something through the cell membrane. Actually, it brings lactose through the cell membrane. Well, that makes sense. If you're going to utilize lactose, you have to get it in. So the first gene The LAC-Y encodes this enzyme that allows the cell to bring lactose in. The second enzyme, LAC-Z, the second gene, LAC-Z gene, encodes an enzyme called beta-galactosidase. And the job of beta-galactosidase is to split lactose. Again, lactose is milk sugar, but it's a disaccharide. So beta-galactosidase splits it into lactose and galactose. Glucose and galactose. Lactose is a disaccharide. Glucose and galactose are monosaccharides. So it splits it. For a long time, it was thought those were the only genes associated with this operon. And then they found a third one. The third gene, the LAC-A, encodes an enzyme called thiolgalactoside transacetylase. I don't make these names up. Okay. Why? I don't know. But basically, what this does is it converts lactose into allolactose. Allolactose is a modified form of the sugar. So this enzyme, thiolgalactoside transacetylase, converts some of the lactose into another substance, allolactose. And that allolactose has a very specific function. It's going to bind with our repressor, and it's going to inactivate it. So allolactose is an inducer. It's going to make the repressor let go of the operator site and turn this operon on. So when there's a lot of allolactose, which means the cell is getting lactose in, taking it apart, and then some of the lactose gets formed into allolactose, it's going to bind to the repressor protein and inactivate it, and then the repressor falls off the operator. I wonder if I had to have an animation of that. Oh yeah, here's our repressor on the operator. Now it's gone. I guess it just fell off. Oh yeah, it's gone because the allolactose took it off. Okay. Now, do you see any problem with this? We've got enzymes that are encoded by genes where the genes can't be transcribed unless those enzymes are made. How are you going to turn this thing on? Ever. It's supposedly off. That means transcription is off. You can't remove the repressor unless you have allolactose. And you can't get allolactose unless you can bring lactose into the cell. So the cell is kind of in a bad situation. However, it turns out that this thing is not entirely off. That is, transcription is never entirely off. Apparently, it's off like a leaky faucet. So it's off, mostly, but there has to be a little bit of transcription of those structural genes, LAC-Z, LAC-Y, LAC-A. There has to be a little bit. Well, if the E. coli find themselves in the gut of a calf, they can't turn this system on. And they need to be able to turn the system on because in a calf, there's not much else to eat. They get milk. So it's off like a leaky faucet. Again, if you want to get into the details of this, I'm sure that you can find more details on what I'm giving you. So this is a kind of simplified version of this whole story. So in order to induce the operon, they have to be able to get some lactose in, and they have to be able to convert some lactose to allolactose so that they can switch this thing on. So you have an operon that controls lactose utilization. It's mostly an operon that is off. That is, transcription is repressed. It's mostly repressed. It can be induced, but only if you inactivate the repressor, which is constitutive and active alone. To activate the repressor, you have to have allolactose, which requires that the cell bring lactose in and convert some of it to allolactose. Now, again, bacteria want to be extremely efficient, so they have another layer of regulation on top of this inducible operon. Because the inducible operon, we just found out, leaks a little bit. It's possible for transcription to happen a little bit. And they really, again, bacteria want to be extremely efficient in terms of energy conservation. So they put another layer of regulation. How many of you have been through physiology? You get the idea in physiology the same way. You've got this that regulates this. Oh, and then there's this that regulates this, and this that regulates, and this, and this, and this. There's backup systems. And that's very, very common in living organisms, where you will have some kind of a backup mechanism to make sure that the first mechanism actually is working the way it's supposed to work. So this mechanism I'm going to be talking about next is called catabolite repression. And we're finally going to get to the point where we're talking about a regulatory molecule that is a nucleotide. So catabolite repression, right there, it's like, what does that mean? Well, repression is gene level control. Repression is stopping transcription. And in this case, we're going to find out that this transcription is repressed because of a catabolite. So then you have to know what a catabolite is. And then we're going to throw in the fact that this involves a regulatory molecule called cyclic AMP. So, what is catabolite repression? Basically, catabolite repression is a mechanism that is going to let bacteria like E. coli use constitutive enzyme systems in favor of inducible ones. In other words, use enzymes you've already got. If they're constitutive, you can't turn them off. So if you have an opportunity to use constitutive enzymes, use those first and use inducible ones second. And catabolite repression is going to make sure that works, make sure that's how it happens. It's kind of like if you have children at home and they hear the ice cream, the jingle of the ice cream truck, and they're all wanting to rush outside and buy ice cream at the ice cream truck. And you say, no, I'm sorry, we have ice cream in the freezer. You're not going out and buying something new because we've already got ice cream. E. coli is telling their kids that all the time. And no, you're not going to go out and use some new enzyme pathway. You're going to use the enzyme pathway you've got. Okay, so it's just, again, it's a second layer of regulation that is exerted on an inducible pathway because the inducible pathways are leaky. So, what is a catabolite? A catabolite is anything that the cell can break down, catabolism. If they can break something down and use it as an energy source, that is a catabolite. So the one that we talked about most extensively is glucose. Glucose is a catabolite. It gets broken down. If the cell is a respiratory organism or a fermentative organism, typically you start with glycolysis. So you have the glycolysis pathway and then you can follow that with other pathways. Anyway, so glucose is a very important catabolite, and it is going to exert control over inducible operons in lots of different bacteria. Typically, the enzymes associated with glucose catabolism are constitutive. So the enzymes of the glycolysis pathway, for example, usually those enzymes are constitutive. If you put E. coli into a medium that has different sugars, and we have TSI we use in the lab, TSI, triple sugar iron. That medium has glucose, sucrose, and lactose. The bacteria will use glucose first. They'll use lactose last because the lactose operon has to be induced. And the operon that regulates the enzymes necessary to run the glycolysis pathway, those enzymes are constitutive. The operon doesn't have an operator site on it. So if the cells can break down glucose, they will. They're going to eat glucose before they eat lactose. This is a very simplistic diagram showing glucose being catabolized to make ATP, and then the ATP being catabolized to release energy. It's kind of handy, except that it's not very accurate because cells don't actually make ATP using adenosine monophosphate. They make ATP using ADP. But I'm trying to, in that diagram, I was trying to show you that AMP is going to have a role in this. It's actually cyclic AMP, and it has nothing to do with that diagram. The only reason this is in here is because AMP is going to be our regulatory molecule. And if you look at that diagram, if glucose is being catabolized, ATP is being made, so the arrow is pointing upward to make ATP. At the same time, if you're increasing the amount of ATP, you're decreasing the amount of AMP. So you can help remember that if the cell is actively utilizing glucose and making ATP, there's going to be very little AMP available to be the regulatory molecule, cyclic AMP. So it's handy from that standpoint, even if it's not accurate. reason again, it's not accurate. So it turns out that when glucose is being catabolized, cyclic AMP levels do drop, but it's not because of that diagram that's shown at the top. It's because when cells are actively utilizing glucose, they, for whatever reason, stop producing an enzyme called adenylate cyclase. And adenylate cyclase is necessary to make cyclic AMP. So if they're bringing glucose, and it has to do with bringing glucose through the cell membrane, when they're bringing glucose in, it inhibits the activity of adenylate cyclase. And again, adenylate cyclase is the enzyme that makes cyclic AMP. So if they're getting glucose, they're not making cyclic AMP. Well, so then why does that matter? As I said, cyclic AMP is a regulatory molecule. It's going to regulate at the gene level, so it's going to regulate transcription. It turns out it doesn't do it all by itself. There's some other molecules associated with it. One that you need to know about is called catabolite activating protein. So cyclic AMP wants to bind to DNA, but it can't do it by itself. Typically, DNA is not going to be bound by a single nucleotide, but it can be bound to a nucleotide that is associated with a protein. So cyclic AMP interacts with the protein, catabolite activating protein. This is sometimes referred to as the CAMP complex, where CAP is catabolite activating protein, and cyclic AMP is just CAMP, so it's CAMP. On the other hand, I hate people reading acronyms as though they were words, so it's really catabolite activating protein as in CAP, and cyclic AMP as in CAMP. Anyway, they form a complex, and that complex binds to DNA, but it does not bind at the operator site. Instead, it binds somewhere near the promoter, and when it does, it's like, here, this promoter is really exciting looking. Why does that matter? Well, because, again, the promoter is where sigma factor binds, and it turns out that for inducible operons, the promoter sites are weak. They're not attractive to sigma factor. Apparently there's, as I said, there's different sigma factors, but the sigma factors that would normally bind to these inducible operons won't unless those promoter sites are enhanced. You have to make them more spiffy looking. Notice it's more spiffy, especially if you were watching the fireworks before it got to that point. So the binding of catabolite activating protein and cyclic AMP makes that promoter more attractive to sigma factor. Consequently, now sigma factor will bind to that promoter and go ahead and initiate transcription of those genes, assuming we've already gotten the repressor off the operator site. But again, this is just another layer of regulation on top of the induction. So if that promoter site isn't enhanced, then typically sigma factor won't bind. And even if you don't have a repressor on the operator site, transcription is not going to happen. You have to have sigma factor bind. Make sense? Kind of, sort of? Maybe later? Maybe later when you have a chance to think about it. Yep, gotta make our promoter site more spectacular. Okay. Anyway, again, because the enzymes that are involved in glucose catabolism are constitutive enzymes, you can't turn those on or off. Typically the cell's going to use glucose first. And if they can use glucose, they're bringing glucose in and breaking it down, then they can't make cyclic AMP, and you're not going to enhance that promoter site on an inducible operon. And it's not just the lac operon that works this way. Although, you start looking at operons, there's a lot of variation on the theme. Yeah.
So inducible operon only works when the catabolite repression process takes place? I'm trying to wrap my head around this.
So it's not going to work if catabolite repression is in place, because catabolite repression is glucose, the catabolite is repressing transcription. As long as glucose is available, they'll use that. And that means you're not going to induce that operon. You have to get rid of glucose so that you're not having catabolite repression. And then the cell can make cyclic AMP. and then you can enhance the promoter site. So it's when glucose isn't available. So again, glucose, the catabolite, represses transcription of inducible operon genes because it blocks the production of cyclic AMP. The cell is bringing in glucose, it doesn't produce adenylate cyclase, and then it can't make it cyclic AMP. Without cyclic AMP, you can't enhance the promoter. There's CRISPR information. Woohoo! This is just another look at this whole CRISPR thing. I talked about CRISPR gently before, or briefly before. You guys are hating this though, huh? Maybe we should just stop.
Yeah.
Let's just
Let's
stop.
just stop. Please.
Really, because the operon is like this one layer of regulation. Somebody was just asking me today, has microbiology really changed that much in the last 10 years? I just go, oh my gosh, you have no idea. It's out of control, completely out of control.
Oh,
And
it's out of control.
it's extremely challenging to teach because you don't know what to include and what to say. Never mind, we're not going there.
Or what's different.
Oh, yeah, it's just awful. Changing so fast, it's really mind-boggling. Okay, next time we're talking about CRISPR.